we'll go over the answers for the bioinformatics tool on the reverse complement. So this part here is asking how many base pairs, and basically you just count, you could count how many pentoses there are, or you could count how many nitrogenous bases there are in like either this strand or this strand. So DNA is double-stranded. So this guy here is called the phosphate backbone. So this was like, so this is the one strand, and there are these guys sticking inward, and they bind to the other guys sticking inward from the other strand. So how many base pairs? 1, 2, 3, 4. If this was RNA, it would be very similar to this, but it would only have, let's say, the left half or the right half. And it would be broken right here in this part where it's called the hydrogen bond. So it would be broken right in here, 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 and here. The other difference, if this was RNA, there wouldn't be a T, there would be a U. So it would be an uracil instead of thymine in here. And let's see what else. Also for RNA, whenever we count RNA, we count how many nucleotides. So this is one nucleotide. This is another nucleotide. And because the RNA doesn't, okay, actually does form some pairing, but let's pretend it doesn't. So we just count the how many nucleotides. DNA, sometimes you say nucleotides, but we also refer to it as base pairs because DNA occurs as double-stranded. Sometimes it's single-stranded, but just think as a general rule, DNA is double-stranded. So it's always married to somebody. And RNA is almost always not married to somebody. It forms pins, which we'll see later. Okay, then I asked, what are the 4 sequences you could have? And you're supposed to read from the 5' end, which means you're reading the base that is attached to this pentose. And so you're going to read this way. So the 5' carbon is here. This end here is this is called the 3' carbon. So we read 5 to 3. That is the convention of reading DNA. If you read from bottom up, so we read the bases, then you'd have to write 3'. So you'd have to write 3', and then the sequence, which would be, okay, in this case, G, pink is T, C, and A, 5'. Okay, so this is to indicate I didn't use the convention of writing DNA. The convention is read 5' to 3'. So if you used the convention, you could write just letters, which is what we normally do because we understand that this means that I'm reading 5' to 3'. So this, if you look at a DNA sequence that is written like that, you can assume that it's reading 5' to 3'. When they are ordering primers from a company, so we could actually say, I want this chunk of DNA sequence. You type it in on a form online, and they make the molecule for you. And many times the form will say 5', give you a bunch of spaces, 3', because, and you fill in which bases you want in between. They want to make sure that you are writing in the correct orientation. So normally people that are ordering the DNA, they know it's supposed to be 5' to 3', but the company doesn't want to be responsible for your reaction doesn't work, and then they're gonna think it's them, but really it was you read the other sequence. All right, so this part here is probably the part that is hardest for people that don't know about the central dogma of molecular biology. So let me go over this part maybe in the most detail. Okay, the blue is a DNA strand. And if it's being transcribed, then this part of the DNA is a gene. So remember, genes are DNA sequences that are transcribed. And here it's making some RNA. We don't know if this is messenger RNA. If it is messenger RNA, then that molecule will be read by the ribosome, and there will be a protein molecule made out of it. If this molecule is a ribosomal RNA, then it doesn't get translated. It just exists as ribosomal RNA. So there are genes, DNA sequences that are transcribed into tRNA and stays as tRNA. So those are called RNA genes. But whenever people think genes, they think, oh, DNA sequence that encodes a protein. And then I'll say, okay, most of the time, that's a simplistic way to look at it, because there are DNA sequences that are not translated to protein, they just stay as RNA. And if I forget this, it's fine. There will be another class on gene regulation, and I'm going to repeat this. So, all right, so let's see what else about this. So the blue is the DNA sequence. This orange blob here represents an enzyme, which is an RNA polymerase. So the A's in molecular biology indicates enzyme. Polymer means some structural repeating unit. So in this case, it's repeating, like it's a nucleotide, another nucleotide, another nucleotide, another nucleotide. So it's the repeating nucleotides, right? They don't have to be the same, but it's a bunch of nucleotides in a chain. So, and the RNA polymerase, because it's an enzyme, make polymers of RNA. Okay, so this enzyme, what it's doing, it's reading one of these two DNA sequences. At this point, I haven't told you which one, and it's making an RNA molecule. So in the genome, you will see later again, each gene is the RNA polymerase. It's either going to read this strand or this strand. It doesn't just decide on the moment. There are cues in the sequence for the RNA polymerase to know which one it's reading. So we'll learn more about that. But let's see what else about this. So in this case, it's saying the direction of the synthesis is from 5 to 3. So when we say 5 to 3, what we mean is it's adding nucleotides to the 3' end. It's reading this strand and saying, okay, I read an A, so I should put a U there. So for RNA, there will be a U. If this was the DNA polymerase, it would be putting a T here. And then it reads a C, and then it says, I should put a G there. So what it's doing is reading the sequence, making this molecule. Okay, so it reads from 3 to 5, but it builds 5 to 3. Okay, so some names that are important to know is, for example, template strand. So it's called a template strand because the RNA polymerase is reading this strand. So this is the template it's following to build this one RNA strand. So template strand, not the template strand. So it's actually not reading this, it's ignoring this information. And let's see what else. If you compare this sequence here of the RNA with the non-template strand, so let's do that. Same thing. TU, not the same thing because it's RNA, so it puts a U instead of a T. And then GG, CC. So if you are not convinced, look at this whole thing, and then you're going to see that this pink sequence here is the same sequence as this blue one. So the messenger, so this is messenger RNA, so the message here is the same sequence as the non-template. Okay, so normally I will ask you, what would be the sequence of the mRNA if I gave you this template DNA? So then you go like, oh, the sequence is going to be something that is complementary to this. So, and if I gave you the template strand, then that would make it a little bit hard, because this is actually how we do it in the lab. Remember how we write sequences from 5' to 3'? So I would write this sequence here, but I'm not going to read from here to here. I would give you this sequence read from here that way. Initially, it's very confusing. Your brain goes like, oh, it's doing this, cartwheels. But if you stay in this field, eventually you're like, yeah, sure. So I would write this sequence. I would be writing it from left to right. I would write TAC. So I would be reading this, but writing the other way, right? So, because that's how the convention is. So I would give you this sequence here and say, this is the template strand. What is the sequence of the mRNA? And the answer would be this guy here. And so you will see that this sequence here is what we call the reverse complement of this sequence here. So maybe let me back up a little bit. If I just read this as it goes, like AUGCC, I'm reading it in the conventional orientation, that is 5 to 3. So that's fine. And then A is complementary to T. So this base here is complementary to this. This base here is complementary to this. So if you just give me the complement, you have to write 5', then this sequence, which is the complement of this, and then you have to write 3'. Let's check if you're paying attention. So you write 3', you write this sequence, which is the complement of this one, and then 5'. But normally I want you to tell me in the conventional orientation, so you'd have to flip it. That's why it's called the reverse complement. So if I read this sequence here, I need to go from here that way, and then I would have to write the reverse. All right, so this is probably the hardest part of this. So the names to remember are template strand. Then it's easy to remember the other one is not the template. Normally I just say either the messenger, so or what is the mRNA formed out of reading this template strand. Or maybe I say, here's the DNA sequence of the non-template strand. And then I ask you, what is the mRNA sequence that corresponds to that non-template strand? And it's basically the same thing, just changing the T's for use. All right? So let's see. And you can see all the answers to this when you do your first submission. I think that's mainly what I wanted to go over. And some of you might have submitted your answer in this box, and you're certain about it, and you considered it wrong. It might be because you put a new line character somewhere, and so you got like some sequence, a new line character, and then another sequence, so like it looks like another paragraph. The computer reads that as 2 words instead of 1, and then considers it wrong. When it's exam time, what I do is I go over every answer that was entered, the computer says it's wrong, and then I see, is it really wrong, or is it just, one copy and paste, one letter missing or something, or one space that was added, and then I regraded that. All right, so I think mainly I wanted to spend time on this because of people that don't know about DNA conversion to, or making mRNA based on a DNA sequence. Are you guys good with that? Do you have questions about that exercise? You okay? All right. Thanks for responding, Molly. So, all right. Thanks, Taylor. So let's see. I'm going to go over now the PowerPoint presentation for genome assembly, and then you're going to do the exercise. So hopefully you have these guys with you, and it should make it more fun. And if we don't get to the end, that's okay. We'll pick up where we left on Friday.